So, hi everyone. We are very lucky to have Thomas here, Thomas Chartier from Marseille, who decided to make us the pleasure of coming to Paris to give us a research seminar. So, a lot of us know Thomas from a long time. We met where we were both with Vincent, Elena, Ernest, AIV students in the master degree. And Thomas took, after the AIV, a position at the MBL to do his PhD on, I would say, neuroscience of novel model organism, maybe, something like that.
Good way to
and then got a Marie Curie Fellowship to go work in Marseille and study learning associations, or yeah, you could say something like this. And Thomas was visiting us a few months ago, and pretty much so we're working, and offered us to speak about the topic he will discuss today. And we said it would be even more fun if, instead of giving us a seminar just to our small group, we would open this to the whole CRE. And so we are very lucky that he's doing that today. So thank you for coming, and yeah, the floor is yours.
Thanks, Owen. Yeah, so the talk was meant initially to entertain the MDM lab, indeed. So hope it will entertain you as well. You may learn something. So it's not so much about what in the research, but rather about how we do it, right? And I think it's interesting from time to time to also discuss how we do it. And as you will see, this works for all kinds of disciplines. So it's really not specific to what I'm doing, which is cognitive psychology. The starting point is that we are really full of expectations. So the brain is predicting at any moment what will happen, what will not happen. We expect so many things that we are not aware of. For example, if I walk here, I expect that the floor is going to stay solid, it's not going to fall. You expect that your chairs are not going to collapse. We expect that there's not going to be a loud noise right now. We expect that, I don't know, there's not going to be a pandemic striking down on world economy. Well, we expect all sorts of continuity. So we continue to make assumptions, and we have to do it because otherwise the brain will be completely overwhelmed with information. So we say, I know this, I predict the rest of it. And that's a source of a lot of errors, a lot of distortions about reality that I will now illustrate with a couple of examples. So I'm going to talk about cognitive biases, but more generally about how, like, the interaction between how our brain works and what happens in the process of research. Okay. So let me start with an example, probably outside your field, I guess. It's about research in dolphin vocal communication. So I saw this seminar recently, and dolphin communication is very rich. They have all sorts of vocalization, they have group behavior, they are very social animals. And it was studied so far with group recordings, meaning that you would put a hydrophone under the water, record the scene, visual and audio, and then you will say, okay, this dolphin is making this noise at this moment, and you will make a lot of conclusions based on this, conclusion about social interactions and what they can say. But actually, they realized recently with a new technology that allows them to pinpoint exactly where in space the sound comes from, that the dolphins who are speaking are not the one they thought. Because they had made assumptions that, for example, this dolphin is being more active now when I hear the vocalization. So I assume that he's the one speaking. And now with this new technology, they see that a lot of those assumptions were actually wrong. And Fabienne Delfour, who's working also at CNRS in Orsay, told me that they have to completely rethink all their conclusions, basically. And they have to acquire the data with this. Okay, so there was a sort of chain of assumptions, and not all of them were verified. And this one was really a missing link. So that was, you can say, really an expectation bias. Okay, people expected, oh, this dolphin is speaking right now, And there were a lot of anthropomorphic, anthropocentric projections onto the dolphin behaviors. Right. So cognitive biases, well, that's a classification of them, 188 in Wikipedia. So I'm not gonna go through all of this, obviously, because you would remember nothing. I'm just gonna pick a few instead. And the important thing is not to know all of them, but just to think about that, like get used to think about that in the way you're doing science. So that's a depiction of the process of science, you know, this cycle of generate hypotheses, we test them, we see if they were correct, and then we regenerate new hypotheses. Okay, so now I look like a consultant with a graph like that. So we all know it's really a bit messed up, like you have some blurry thoughts, you do the experiment, you communicate your results, then maybe you analyze, so then you need a hypothesis, then you generate it, and etc., etc. But so I'm going to discuss a few steps in this process. And one first example is what I call looking at the wrong place. And I really like this one because it's as easy as it can be devastating, or at least it can really delay discoveries in science. So the example I'm taking is that of rock paintings in caves. So you know this, I guess. You see the hands that are painted on the wall here. Yes. So you would probably expect that these paintings were discovered by just randomly discovering a new cave. You enter, you lit up your torch, and then suddenly you see the paintings. That's really not how it happened. Actually, in this case, so the Gargas cave in the Pyrenean mountains in the south of France, people have been searching the cave for 30 years. And it was not until then that they discovered the paintings. Okay. So the reason about this is that they were looking at the wrong place. Because at that time, they had found some bones of animals that were in between chimpanzee and a man. There was this idea of maybe our ancestors, prehistoric man. We had found some tools, and people were looking for a missing link that was more an animal than a human. And they were looking accordingly on the floor for animal traces, okay, remainings that would be buried on the floor. And they were just not looking up to the ceiling, which was more a sign of spiritual elevation, something cultural. So that's not what they were looking for. And then suddenly, after 30 years, this person randomly sees that on the ceiling, and this has completely changed the way people were seeing our ancestors. Okay. So what I like in this is that the limitation was really not technological. You know, it was not about having the right lamp, having X-rays, having whatever to detect the thing. It was really cognitive limitation based on the assumptions they are made, based on the expectation. And that's something that we can see quite often, and maybe you will find yourself examples of that. So I'm giving you a neuroscience example, which is where in the brain the words are represented. And there was this logical assumption since the 19th century that words would be represented in language-specific areas. So we have known with Broca, Wernicke, all these neurologists, that there are some areas when they're damaged, then language functions are impaired. So it was a natural assumption that words would be localized there. And actually, people around Friedman, Pulver, Müller started having a different assumption made on a different theory. And here's a nice paper where they show that action words involve the motor cortex, okay? The part of the brain that is programming the actions that we're doing. And it kind of makes sense, because when you learn these words, these action words, initially it's probably in moments where you're also doing the actions yourself, right? But it depends on the theory you have behind. So people have been looking on the floor for a long time. Localization is approach. It's language. It's a language area. But with a different framework, in this case, Hebbian learning and an associationist framework, then it makes sense to look in what is associated with the learning of the word. So in the case of action word, motor areas. And when you start looking, suddenly you see it. And people could have looked at that much earlier, just pin an electrode in a motor area, you would have seen it without much technology. So the same could happen for you, like if you're looking for some genetic mechanisms, phenomena, like the old version would be you would look, one gene gives one function or one trait, so you look in the coding genome, right? But now we know that there's a lot to find also in non-coding DNA, so maybe people start to look up. And another example could be for if by chance you're looking for a next antibiotic, right? You could look in bacterial diversity in the soil. That could be a good place to look up. Anyway, so just keep asking your question. That's it. Are you looking at the right place? Did you make assumptions that are limiting the places where you're looking? I think that's particularly true for DNA, right? So that was the looking at the wrong place. I think it acts mainly on hypothesis generation, but not only. So now reading exercise. Can you read that for me? Whoever is able to read it, please raise your hand and speak up. Should be able to do it.
Learning, research, collective intelligence, and adaptive.
Okay, learning, research, collective intelligence, creativity, and help adapt to a rapidly changing world. Does everyone read that? No? Yeah?
But yes, no, that's not what's written.
That's not what's written. Yes. And actually, you're not reading, right? You're not doing what I ask for, right? So you are projecting things that you know. You want this data to be what you say, but it's not exactly what it is, right? It's a character string, like it's a research co-clave intelligence creativity knar hepat that repladik na hagning word, right? That's the that's the raw data. Okay, so here, of course, you're seeing a pattern that there is to see, right? But your brain is projecting this.
But it's not like we were seeing the words written in the correct order. So
What do you mean?
it's not that we didn't do what you asked us to do, it's that we interpreted what you asked us.
Yes, exactly. So you assumed something, right? You assumed that I was expecting you to find the English meaning in a sentence, or you assumed something else?
I'll see where you're
Color of the balls. I mean, I guess probably you know this one. So what color? What do you see?
Red,
Okay, red, purple. Well, different colors.
copper.
Okay. So it's also a question of how far you are from the screen. So as you would have guessed, the balls all have the same color, like in all these perception tricks, right? But if you're a bit further, your brain will interpret them as red, green, blue. Okay. So here in this case, it's seeing something that there is not to see, right? The reality is that they're all the same color. They have this copper or beige, and your brain is interpreting something. So that's sort of the opposite. So that's what I call not seeing versus seeing something else. Another one now, I just ask you to not to say the thing if you see it, not to bias the others, okay? So that there is a pattern to see in the next slide. And please just raise your hand when you see it, if you see it. So I guess some of you will know the picture. Okay. Some people see that. Not up. The rains go up and down. Okay. So now, so for the people who haven't seen it, if I now tell you look for a dog walking to the left with the head in the center of the picture, do you see it? Like, does everyone see it now? No? It's a small head. The dog is sniffing on the floor, walking to the left. It's still hard to see, but don't worry. So that's the head. The legs are here, front legs, back legs. Okay, so that's a Dalmatian dog. Do you see it now? Yeah, okay. So there's this raw data. Some of you see the pattern. Some of you need a bit more information, maybe. But that doesn't matter. It's just maybe dependent on what you have in your brain at this moment, or whether you're used to seeing dogs or whatever. So it's just about, do you see the pattern, right? And now that you've seen it, next time I show you the picture, you will see it, right? So when you've recognized the pattern, it's much easier to see it again because the brain knows what to look for. And if I now show you the picture upside down, you will say, yeah, okay, I kind of see it. Like if you have seen it like this way at first, you will never have seen the dog, I guess. Or if you see partial data, you will also find it, right? So once you've seen the pattern, it becomes much easier. And I think a lot of this describes scientific discoveries, not all of them, of course, that it's difficult to see the pattern, the Dalmatian, for the first time. But once someone else has seen it, for example, you say, yeah, obviously, why didn't we discover this before, right? So an example of this, maybe it's evolution of species, like something we had under our eyes for a long time, but we needed to learn to see these things, right? To think differently, project something different in the data. Giving you a quick example from my PhD, where I had something under my eyes for 1 year and I didn't see it because I didn't know, I was not able to recognize the pattern. In this case, was looking for neuronal activity in those tiny brains. So that's 200 micron. It's a head of a tiny marine animals that I was working on, plantaries. And I was looking for activity in potential learning-related areas. And actually, I had seen it in my very first recordings, calcium recordings that I acquired, but I didn't know what I was looking at. And not until I did precise neuroanatomy to know what I was looking at, was I able to understand that actually my discovery was already made, right? So, and ironically, the best data I had was from the first recording session. I never got that good quality afterwards. So, learning to see. Giving you another example of seeing a Dalmatian, so that some of you have read this book. This is an amazing book from Sean Carroll. It's about the genesis of like modern gene coding theories with Jacques Monod, François Jacob at Pasteur Institute. And here's what François Jacob describes in 1956 or so. He says, suddenly a flash. Suddenly he sees the Dalmatian, the pattern in the data. How I didn't practice that one. How could I not have thought of it sooner? Both experiments, that of conjugation, the phage, so it was some floor in Pasteur Institute, and that done with Pardy and Monod on the lactose system, another floor at Pasteur Institute, are the same results and conclusion, okay? For years, no one had seen any meaningful connection between the two. So suddenly he realized, okay, we're working on phage multiplication, but it's actually exactly the same thing that's happening as with the synthesis of galactosidase. And he's connecting the two by this, he sees the common pattern, the Dalmatian, if you wish. In this case, a gene that codes for a repressor, right? So at that time, that was a big conceptual leap, as you know. So yeah, that was about not seeing the pattern or seeing a wrong pattern. So that's when you analyze your data, when you see the raw data, right? So look for the Dalmatian, but when you see one, ask also yourself whether it's real. So like for the balls, you see different colors. Is that real? Because our brain wants some things to be real that sometimes are not. And this leads me to the next cognitive bias.
Oscillating between both, like did I meet something or am I seeing something that is not true but I want to see? And beyond the fact of keeping that in mind, are there like techniques to kind of hack our own brain so that it goes one way or another? Or how do we tackle that pretty much now that we know this bias? Like, do we have a way to tackle it?
I would say an answer can lie in the next bias, so confirmation bias. So what? Yeah, okay, I'll answer. So confirmation bias, you know, right? We want things to be true because they fit our theory, our hypothesis. So that's a recent example. After the debate for the French presidential election, so between the two rounds, you say who was the most convincing in the debate. You ask Macron voters, they say it was Macron. You ask Le Pen voters, they say it was Le Pen. Okay, so the opinion poll is pointless, right? No one is judging the debate objectively, and everyone already has the answer because before the question is asked. So that's an illustration of confirmation bias. So when we want to demonstrate something in science, we should really remember it's like failure to disprove, right? We should try to prove the opposite. And if we keep failing, proving the opposite of our hypothesis, then we should say, okay, then I have to accept my hypothesis. Damn it, right? And you cannot get rid of the signal. So that's why our opponents are the ones that test our ideas better, right? Because they really want them to be wrong. So they do all possible experiments that we're a bit lazy to do, maybe. And if they really fail, then probably it was a good hypothesis. And if both have some truth, then probably none of them is good, and you have to merge them or like, go beyond. Yes?
So it's an absolute necessity that the hypothesis is falsifiable.
Yes. That's proper, yes.
Because you can have a failure to disprove, but no way to disprove, but no way to disprove it actually. Right? Like, I mean, we have a long conversation about flat earthers here. And right, like someone who says that the earth is flat, I mean, we have ways to prove that. Or like, no, God exists.
Yeah, existence of God is the canonical example. Yeah, within the framework of falsifiability, that's proper described.
Was more about building on your hypothesis of against your bias, but not seeing something and trying to disprove it because somehow you are not ready to take. I fully understand that for the confirmation bias, but sometimes it's about kind of saying, okay, maybe I'm wrong about what I'm expecting in my hypothesis, you know. So I don't think it's necessarily.
Well, here I'm talking, so for the neuron example, it's more once I've seen the activity for the first time, then I really should try to disprove it, right? But as soon as you have not seen something, you know, absence of proof, right? And so to answer you, how can we tell between not seeing something or seeing something wrong? We have to ask what are our hypotheses, what are our expectations, and this pattern that I see, does it confirm what I want to be true? And if yes, then I should be very careful. And I should really try to disprove it. And do some of you do pre-registered studies? Like, have you started doing that? No, you know about this? Yes, not many. So it's like your method and your hypothesis are peer-reviewed before you acquire the data. Okay, so the journal agrees to publish your results, whatever they are. And in this way, you don't change your interpretation, you don't redo experiment, you don't remove data, right? So it's a recent way of publishing that has emerged, and it is peer-reviewed. So that's and apparently some studies prove that it's improving those issues of confirmation bias. So yeah, check it out.
But then I'd say also, you know, it really assumes that, you know, like science goes from, you know, hypothesis
With,
A to result B,
yeah,
and that we're not actually in the process of finding C, you know, which is, you know, I don't have numbers, you can't really quantify how many projects actually end up exactly where they wanted to end, but I'd say it's a minority.
Yeah, that's true. That's a big problem with those pre-registered study, that you will want to maybe trash your experiment where you invested already time on having it peer reviewed. Yes.
I guess that's a good way to settle a debate. Field has built debate going on. You propose one set of experiments, and if everyone agrees on the set of experiments, then you're put up, right?
Yes.
So it sort of fits a very specific kind of research.
Yeah, I think it's good to include them, but I don't think it's reasonable to have just that, because it's too limiting, in my opinion.
I mean, at the same time, I don't think they prevent you from publishing identical C, you know.
Well, you really have to think in advance of, and if this doesn't work, and then there's this other hypothesis, and you really have to imagine everything, which you can't.
Biology, it's not very much like physics and astronomy.
No, when in our field, psychology, we have this, like neuroscience in general. I don't know if it's all over the place. I don't know that much about it, actually. So here's an example, confirmation bias. So that's a paper that I recently published with my boss, Joel. So what we are working on is a comparison of cognition between non-human primates, so baboons in our case, but it could be chimpanzees, could be bonobos, macaques, and humans. And there's a huge expectation for some of the researchers. They want to see a difference between humans and others, right? And this idea of the scala natura is still very important in the field. And so we have a particular question, a particular cognitive trait. It's about the symmetry of association. Doesn't matter that much that I describe it. The idea is that humans have this capacity and the other species don't have it, right? So I looked at actually the human literature, and I found that the proofs for this phenomenon are actually right, almost zero. Like, I took 37 articles, I reviewed their method, and really like 4 have a suitable method, and 1 has convinced data. So I'm not saying that it does not exist, but it's completely overstated. Right? And why is that? It's because people expect it to be true. So they don't care about false positives. They don't verify the things. Whereas on the other side, when we find traces of it in non-human animals, we are super critical about the data. The reviews are absolutely horrible. Like, you need to run 10,000 controls. So there's a complete discrepancy between the 2 expectation levels on both sides, right? So this is basically what we're saying in this article, and the reviewers were really positive about that. So confirmation bias, I would say, mostly when you interpret the result, but also when you generate hypotheses, like, so it's very common. Yes?
All of this confirmation, all the bias you're describing, I see it a lot, something I'm thinking about is the role of a lot of, when we talk about sexism and inequality, people think cannot be a human, but a lot of it is confirmation bias or the type of question you're asking. And typically, research on, I don't know how you learn about reproduction in school, but usually it's a passive oocyte and a spermatozoid that fights with a lot of other spermatozoids, a super dynamic war to reach the oocyte that then we manage to win. And how biased is that? And that's at least how I learned it. I don't know if many of you that's how you learned it. And then it's like the same thing is about kind of active differentiation of the main phenotype and like female by default. So people look for like
Yeah, that's a good
And
example.
for me, this is exactly because people have been projecting their sexist type of stereotypes on the research, and then the research completely influenced by that. And people are finding a gene that directs how you become a man. Yeah, it's normal, it's an active process. But
Yeah.
then they didn't even ask.
That's a nice example because it's projection of like social interactions, culture onto cell biology and genetics. Yeah.
It's now maybe obvious that it's not the case, but research has been really impacted for decades on this subject that there is this kind of stereotypes.
It's a nice example. Now for a different type. So I'm gonna play a little rhythm for you and try to identify the song, right? So you ready for that? I guess people online can also hear it. I don't know if they can speak or, but again, please just raise your hand if you found it and wait for the others to identify it, right? One. 3. Yeah, you have it? No. So people who are smiling really get it. And don't worry, it's really the statistics, it's really hard to get. So
you're asking us to...
I'm playing a song in my head, I'm just doing the rhythm.
And we need to know what song you're playing.
Yes. Okay. Yeah, should I redo it?
Yeah, if you want to, I can do it.
You have it. No? You have it? Happy birthday. Right, so you all know the song, I guess. But so there was an experiment done that shows that like half people would expect. Sorry.
Also the American anthem.
So half the people statistically would expect that you would guess, but in fact, only 3% of people, and so here in this case, just 3 of you in the room found it. So that's an illustration of transparency bias. Like, as a center of the message, I think it's obvious, and I sing in my head, right? But you don't get it. You don't get my message, and I'm surprised, right? So yes, well, in your opinion, what's the biggest obstacle to science? Just like propose.
Out of the ones that you have just...
No, just in general. What's for you? What's the biggest obstacle to science?
Money.
Money. Okay. What else? Money. Okay. Yes, there was another. No. Okay. So, in my opinion, it's not money; it's language, and I'm trying to convince you by a few examples. So, suppose you want to communicate your locker's code to someone, right? With this 10 digits or so. So there are different ways you can say this, like pass on this simple information. Suppose I say it in German, right? So I guess not all of you speak German. So like, do you understand it? Like, not all of you, right? If you don't speak German, that's an abstract character string, right? It has no sense. It contains the information, but for you, it has no sense, right? So I might as well write this in Khmer, right? Like, as if I was a Cambodian person. It would be the same, right? If I say it now in English in full letters, so now you get the information, but it's still not very direct way of communicating this. It's still abstract. It's a character string. So still Khmer, right? If I write the digits, it's more direct, but it's still not everyone uses the Arabic numerals, right? It might still be abstract characters for you, like still Khmer. If I now do this, like I show you a picture, I show you where you should touch, in which order, this is maybe not the most direct, but rather direct way of describing the pattern, right? So that's why, I mean, you know, we say a picture speaks 1,000 words. Un bon dessin vaut mieux qu'un long discours, so that visual communication can be way more effective than language, right? So again, think about this transparency bias. Maybe it's clear in your mind what you want to communicate, but maybe for the reader, it's Khmer, right? It sees the text. The reader is tired, is reading your article. It's just abstract characters. The message is not going across. So when you prepare all these types of communication in science, like ask yourself, so you just Google Translate everything to Khmer and see what's left. So that slide is terrible, right? Look, that's not direct communication, right? So maybe you can keep this example in mind when you're trying to communicate. So much for the transparency bias. Right. Have you been struggling when you're writing a paper, like how should I put this? Ah, no, I'm rather going to make a figure, or I have 8 figures already, that's too expensive. Like how to formulate the things, right? Now another one that affects decisions that take place at all moments in the process. This is more related to social psychology. It's not so much cognitive, right? So do you know about that? Escalation of commitment? Escalade d'engagement? Not that much. So it's about how you decide about things.
Yeah,
yes?
Like when you keep doing the same thing and then realize that you're wrong, but since you've been doing this forever, you convince yourself that you were doing it right.
Exactly. You know already that it's a wrong chain of actions, and you will continue to prove that no, it was a good decision. And sometimes you invest more effort and money, and time. So you're waiting for the bus, doesn't come. You could walk home, but you still wait for the bus. At some point, you've been waiting for an hour. It was a 20 minutes walk. You know, probably you don't do that, but you see what I mean. So a canonical example is this industrial project, Concorde. So before it was commercially released, people knew it was non-viable because of fuel costs, because of the oil crisis, but still for like prestige and political reason, all sorts of reasons it continued. I have an example from my PhD lab where there was this mutant line that I didn't know what it was. And people were breeding, they kept breeding this mutant line. It was quite some efforts, much more than if you had a Drosophila line with these marine worms, right? And I was never seeing anyone doing any experiment on that. We just keep breeding. It was taking time, money. And then I understood that they just didn't want to abandon it because it had cost a lot of money. And it was actually generated with zinc finger nuclease just 1 year before CRISPR. Too bad, right? And okay. Does it go on pause? I don't know if I should unplug or... No, it's the projector, I think. But, okay, can you try? But there's no light from the projector, so it looks like it's... Okay, well, it's written off here.
Wait, because if you press all the buttons, it takes 20 seconds to...
Okay.
Ça a changé ici ou pas du tout?
Ouais.
Ah.
C'est une vieille tête.
Là, c'est off.
Second biggest obstacle to science.
Right, yeah.
Well, connecting was quite easy. My connection is not stable.
Is that part of the demonstration?
C'était off ici tout à l'heure aussi?
I don't know.
Ok.
J'imagine que non. Ah, there's been a flash. Yeah. Merci. Non, ça va, ça va. I'm just putting full screen.
Trop bizarre.
Non, je pense que ça va arriver.
Ouais.
C'est bon. Okay, so yeah, that was 20,000 euros to the zinc finger nuclease, a lot of money. So that's a typical example. We have invested efforts, money, and that's what is called sunk cost, les coûts irrécupérables, right? So you've paid it already, there's no way to get it back. And because you've paid a lot, you want to get something out of that. Whereas a more rational decision would be to just give up because there were so many problems with this mutant ligand. Anyway.
Are they still picking it up?
I would love to know. I would love to know. I'll ask Flora because she's at Emble now. She will be. Another example is a colleague of mine who was starting a PhD. A project depended on some monkeys arriving from China. It was a COVID lockdown. The monkeys were never arriving. She's been waiting for 1 year, and they were waiting, and she was doing nothing actually scientifically. So I told her, maybe you should change project or quit your PhD, which she eventually did. So it's hard, of course, but you have to accept that it's already too late, right? So what about you? You never had that, right? Like, gosh, I spent so much time on this experiment. That's why actually in social psychology, people recommend that people who put the effort into the things and people who decide whether a project should be continued or not should be separated, right? And I saw my boss recently. I told him, look, we have this nice experiment. My intern had been working for 3 months, working hard on that. He said, yeah, no, it does not make sense. Just remove it. For him, it's easy because he didn't put effort into that. And he's right? Hard to accept for me, but he's right. So be careful, escalation of commitment. That maybe after the presentation, take some time to ask yourself, in this chain of experiment, of action, what about this paper that has not been published for 5 years? What about this person that have been recruiting? Or no, I hope you will not do that, but maybe ask yourself if you're not in an escalation of commitment.
Depends. Like, I know an example of people where I did my PhD. There were 2 labs, 3 PhD students working on a protein called TPS that they thought was a master A regulator of the cell. And then they basically did thousands and thousands of experiments. All the results were negative. And then they did a paper in Cell that said TPS doesn't do anything.
Well, I have a lot of data for that.
Yeah. And then they made the same paper.
It's a good way to save it.
It's a result, right? Like, yeah, well, this thing that people think is involved in other things is actually not.
Okay, nice. So it was not a master regulator.
No, it's not. Yeah.
You're giving me such a good example for another slide.
Yeah.
Thanks. So what was this obstacle again? So I'm actually coming back to this now. What's the biggest obstacle to science? Language. You should not trust language, right? It's very convenient. Of course, it helps to communicate our internal brain states, our thoughts. There are no more slides? Sorry. Sorry, I'm gonna try this again. I should have checked that. Okay. Has now disappeared. Here. No, that's not that. Need one. That should be it. Is it okay now?
Yeah, it's perfect.
Okay, great. So, false friend, French English. Did your boss tell this to you? No. Be careful of a French person speaking to you in English. Right? What's this color? Is it blue or green? Who thinks it's blue? Blue? Who thinks it's green? Okay, well, so it's both, it's neither, I don't know, this language issue. Here, something more profound. So that's the thing that took me most time to understand, because I was trained in math and physics, well, mostly math, and then I switched to biology. That's been actually the biggest obstacle to me for this transition. That in mathematics, everything is complete, is defined, is you know what you're working on, etc. It's true, it's not true, but not all the time actually, but most of the time. When you say always, it's always. There does not exist a single point where it's not true, right? So in physics, usually it's not always, right? It's you have some incertitude that you can quantify with ridiculous precision. You know, it's like really most of the time, and there are a few exceptions because of statistic fluctuation, etc. In biology, it's like, well, it depends on the fields, on the question, but it really took me some time to understand this, that when people were saying always, they were assuming it was obvious that they were like 1/3 of the time it was not true, right? And really, for me, that was was quite hard. And I think that's just one example. But of course, in a place like CRI or in most modern research projects, we have to be interdisciplinary, right? So we have to speak to each other. And that's the big obstacle, because the names in different fields, or sometimes in different offices, don't mean the same, right? So I think we should take time to define the terms. And do you agree on those definitions? Like, do you all have a common definition of a gene? I think it's not that straightforward, not as straightforward as it used to be, right? Or, well, species, we know it's not easy. AI, it's the worst term for this because most of it is not intelligent, etc., etc. And in my lab, we work on associative learning, and my 3 closest collaborators, we don't agree on what it is, right? It's every day we're speaking of things that we don't agree on. So I would say that's an important slide if you want to remember something from presentation. The name that you give to something is just like a sticker on the test tube, something that was put artificially on the reality, right? And you should really remember that it's only a sticker. It will never be more than this. Already between languages, right, it's not the same sticker, so that's obvious. Yeah, maybe sometimes you lost the sticker, so you need to verify what's actually inside the tube, maybe resequence it. Maybe someone else put the sticker, put the name on this phenomenon that you're working on. And if it's a wrong name, you're looking for the wrong things. Well, that was the example by Elena, right? You look for a master regulator. You do all kind of experiment to look for evidence of a master regulator, and it's actually not, right? So you're looking for what I say. Yeah, maybe the tube is empty. Maybe there's nothing under this name. So that's I can give you the example of my postdoc project, where I'm working on the capacity that is called derived symmetry of associations, and now I think it actually does not exist. It was just a name put artificially on something that made sense in a certain framework, but now I'm more and more convinced that this does not exist. So I got funding for that, right? 2 years funding for an empty tube.
But it has a clear definition?
Not really. Every time I speak about this, like when I write my papers, I really have a hard time explaining what it is, actually. That's also how I realize.
They write a paper, but the fact it doesn't exist.
Yeah, I'm planning on that. Experiments. And And maybe you would put a different name now. So I'm taking the example of Weissman-Ids. Do you know this theory of Ids? It was a theory of heredity. So the Ids were thought by Weissman as being the units of heredity, and it was a different concept than the genes. It was sort of popular at that time. Now it's been forgotten. So yes, you're looking for the units of heredity. Now you don't call this Ids anymore because it refers to wrong concepts. Okay. And remember that the name is never going to be the thing, right? Don't identify the name. Don't be too limited by the name. Also because they carry wrong concepts. So a good example of this, I think, is when a journalist is asking a question and there's no good answer. You know, he expects a yes, no answer, something like this. And there's no good answer. And politicians are really good at this. I'm not answering in the way it was asked, right? I have, again, exactly the same problem. In my postdoc, people are asking, are those associations uni or bidirectional? Actually, it's both. So, yes. Name problem that affects all stages, I would say, of the process. Your title, maybe I skip on this. I mean, that's quite obvious. Representativity, you have data for 1%, you claim for 100%, right? So that's very common in my field. People say most animals, they have data for 1 chimpanzee and 20 mice, and they say all animals, right? So it's, anyway, I guess you know this. But for when you're looking for bacterial diversity, I guess you can have this issue. Right? It's very common whenever you do comparative work. You look with evolutionary diversity. So don't overinterpret. So when I put these few biases together, huge mess, right? It's a very crowded slide. Like you don't really remember anything except that it's crowded, right? So maybe you will try to remember that looking at the wrong place is when you're looking on the floor, not looking on the ceiling. Seeing the Dalmatian in the data, seeing this pattern is when you have your raw data. And do you see meaning of that? Are you projecting meaning that is not there based on your assumptions? Confirmation bias, it's very clear, it's very present in science. Think about escalation of commitment. That's, maybe you don't speak so often about this, but it's about your decisions, chain of decisions. And don't trust language, of course. So, that's my summary. Process of science. There are biases. Think about them. And I would say we are used to checking our question, being sure that we have right hypothesis, that the method and technology are the best to address this question. But I think it's also worth investing time on this, like asking ourselves, what are the cognitive biases at play in my project or in this paper that I'm reading? And this can save a lot of time and effort. So yeah, thank you very much. So here are the baboons we are working on. So we are just like 1 minute a word on what I'm doing. That's we have a primatology station close to Marseille in the backcountry, and we have those baboons that are semi-free living, and they have automatized, computerized experiments. So they have touchscreen in free access. It's like a scientific zoo, if you wish, and that's how we test a lot of cognitive abilities in them. Yeah. So they do like it, yes. When so we try not to shut down, but when we do, they are starving for experiments. Yes. So did that resonate with your practice of science? Like, did you identify any biases in your own projects, or yes?
I went from physics into biology, and I was really surprised
coincidental aspect things is so often known, and maybe for good reason, because I'm not sure, but still like that for me to be really almost always as a matter of fact, my first hypothesis that if there's something striking with the gene, the second thing is coincidental, and it's because of physical or chemical concepts, or really always it actually has a second role. So it's really, and I guess, so I have a bias in the biologist, also a different bias. So it's something that
Maybe I should repeat the remark for people online, because I guess otherwise they don't hear the. Yeah. So well, Serge is a physicist working on biology, and the remark is that from a physicist's point of view, when you see an event, you will tend to consider it by default as a coincidence, and maybe then only see if it has meaning. Whereas from a biologist's point of view, your experience is that there's much more projection onto those events, and biologists want to see a function or a meaning in what they observe. That's what you say. Anyone had the escalation of commitment in the project?
I mean, potentially.
Now you can
That,
admit
right?
it.
Like I mean, yeah, as Luke was saying, right? It's really hard as you're doing the project to realize that if you spend 1 year on it. I mean, like TLR, for example, which is a receptor. I mean, it took 20 years to figure that out, right? That it was a very important receptor for the LPS, which is a compound of the cell surface of the bacteria, and it took 20 years to discover the actual molecule itself. So like if we abandon too early, also we are. I mean, how do we know that we're going to abandon too early or that we're going on the escalation of commitment?
Yeah, I think it's difficult.
I would add that the world works in mysterious ways, and it's something like for my PhD project as well. I felt like I banged my head on the wall for 3 years, and then like arrived close to the deadline. Finally, like things clicked, and yeah, I ended up like publishing that thing.
Okay.
And so this sunk cost, maybe sometimes it's not that sunk. I don't know. Like I feel like when you put a plug, sometimes you do miss on things that might unravel mysteries.
Yeah, so I guess the diagnostic. So yeah, the remark is, well, in science we have to insist on projects, and sometimes it takes a long time. So you're giving an example of 20 years of effort until something was found. And S was saying in his PhD 3 years with nothing, and then finally it works. So how do we know if we should insist or not? Maybe a diagnostic feature is when you're sure that you should not. So for the Concorde, for example, they knew they would lose money. Right. So if the reason to continue is to make money, then it was obvious that they should stop. And they continued for other reasons. For the mutant Platineris line in my lab, they knew that there was a problem with an alternative stop codon. There was really a problem that could not be fixed in the line, and it was making data interpretation impossible. So, in my opinion, they should have stopped. So when you know there's something really wrong, then maybe it's a good moment. Otherwise, if you don't know, yeah, I agree. Like, how can you know if it's not going to work at some point? Yeah, in neuroscience, the guy who discovered the place cells in the hippocampus, so these neurons that detect when you're in particular place, and you have a special map in your brain, if you wish. I chatted with him. He got Nobel Prize for that, and he told me everyone told me to stop, and my student was getting very nervous because he had been recording neurons for nothing for the entire time, and then exactly like Ernest, finally, like 1 week before defense, suddenly, he sees place cells, and of course, like they could have abandoned earlier.
Yes. But I feel like, yeah, these stories are more ubiquitous than expected. Like they pop up of the weirdly enough.
But are they rare?
pushing the interpretation of the data.
Well, but maybe they, you know, like maybe you hear of them because they're more interesting than the stories where you know like nothing happens, and so then maybe they're more rare as well. So
No, but maybe also there's like some, in a sense, like when you get close to the deadline, you get some
magic happen. But I would say as long as it's only absence of proof, there's no reason that it's wrong. I mean, maybe it's just hard to find. So it depends on how much you like the subject or how much money you still have, or I don't know. Serge? How does it interface with what? Funding? I know so the question is about like whether we will tend to fake scientific results, to fraud, just because we have been working for so long on the project and investing so much time. So yes, I think, or because we have had a career on this thing and we cannot say, oh no, actually it does not exist. So yeah, well, it happened. I know there are some studies on that in the medical field. I can look up, I have a slide for that. But here in this case, it's mostly about medical diagnosis, where there's a lot of expectation and so wrong diagnosis. How does this interface with funding? I have no clue. I don't know if funding people are aware of that. I think it's really hard to break a chain. I mean, as Vincent was saying, right? A scientific project, it's not going to end up where you thought it would, right? Yeah. And maybe you have to report to your funding agency. I was promising A, and actually I did the opposite because it worked, it was more interesting. So I don't know how tolerant they are to this, and if they can be like educated on that. We should stop.
So we continue. Thomas is here, so we can continue discussing this fascinating topic today and for lunch. So thank you again, Thomas.
Thanks.